Good afternoon, everyone. I would like to thank all of you for attending this presentation today. The seminar will be about environmental DNA, and I will briefly guide you through some concepts, uses, workflows, and some selected results from one of our case studies. Okay, yeah. Although the seminar is about eDNA or environmental DNA, I would like to begin highlighting some of the main motivation behind the fast development and implementation of this technique or this approach in environmental science or natural science. In 2000, the United Nations launched the program Millennium Ecosystem Assessment, and one of the main outcomes of this program was the identification, prioritization, and future trends for a major global driver affecting biodiversity. 5 major drivers were identified, like habitat fragmentation, climate change, invasive species, and overexploitation, and pollution by nutrients. This later, pollution by nutrient, was identified as one of the major drivers affecting biodiversity in the aquatic environment. That study, of course, was based on a lot of research in the early 2090s, and since that moment, a lot of scientific evidence had been piling out, and these categories today have been expanded towards what is understood as heavy metals and organic chemicals as well. But what is clear is that we are talking about of chemicals derived from human activities, the one responsible of threatening biodiversity in the aquatic system. Unfortunately, one of the consequences of societal development is the release of chemicals into the environment, and particularly the aquatic environment. For instance, pesticides enter rivers or streams through runoff from agricultural areas. Pharmaceutical and personal care products reach the surface water mainly from wastewater treatment plant in developing and industrialized countries. Industrial chemicals are present in the environment due to the direct discharge from the facilities, or due to accidental spills as well. And heavy metal, of course, exceeds their background concentration due to mainly mining activities. Globally talking as well, agriculture is quite relevant in certain situation about increasing the concentration of heavy metals, certain metals in the environment. All these chemicals can be detected and measured in the aquatic system, and more importantly, that they are not present as single chemical, rather than forming what is understood as chemical mixtures. Therefore, one of the main motivation behind FRAM, and of course my research, is to define the role of chemical as biodiversity drivers. And here is where environmental DNA is one of these approaches, one of these techniques used to face this question. But what is actually environmental DNA? Maybe many of you have heard about this concept, or maybe you are not, and that is the reason why you're here. But literally, environmental DNA is just nuclear DNA or mitochondrial DNA released from an organism present in the environment. This DNA remain dissolved, for instance, in the water, or can be associated to particles, and then it's transported between the different environmental compartments by mainly hydrological processes. That DNA is also affected by chemical and biological degradation processes, so therefore does not remain forever in the environment. So this DNA is distributed among different environmental compartments, and of course can be also be collected from different environmental matrices, like a freshwater system, like a river or lakes, coastal areas or the deep ocean, sediment from the bottom of the ocean as well, the forest. So literally, you could extract environmental DNA from any environmental matrix that you're interested to. But what is important to recognize is that environmental DNA goes beyond to different group of organisms. So you can extract DNA from microorganisms, passing by invertebrate to big mammals. And then when you have your sample, of course, you need to just use particular extraction techniques in the lab, certain bioinformatics steps, and later on you can have access to all the information about biodiversity. I will briefly later explain one of these steps. But literally, we are in that situation where we have this DNA present in the environment and we can just collect it following certain protocols. The use eDNA is becoming a standard method for biomonitoring. So it's complementing traditional taxonomic approaches today. It's also frequently used for the detection of rare species or invasive species in the environment. Has been shown to be a really useful tool to reach really complicated environment. For instance, caves, a lot of biodiversity have been discovered in caves, submarine caves, and also terrestrial caves. Also has been a powerful tool for algae or algal bloom detection of certain toxic organisms. And of course, what is a bit more relevant for, at least for my research line, that is a really, is becoming a really good tool for assessing the impact of how actually chemical pollution can change certain biodiversity metrics in the environment. There are several studies, there's quite a lot of studies today showing the application and use of eDNA in the context of chemicals and effect on biodiversity. I just wanted to pick up one and highlight actually very nice results from that study. And in this study, actually, the colleagues exposed microbial communities to different treatment with different proportion of wastewater. So you have here different treatment with 0 wastewater up to 80% of wastewater. And then you have 2 biodiversity metric here. One is called the alpha diversity, which can be summarized as a within sample diversity. And also you have another metric understood as the beta diversity, that literally is an estimate for the degree of similarity or dissimilarity in among your samples. So what we can observe here that in this study, they observed changes in alpha diversity at a specific level in a specific treatment. That treatment was having 30% of wastewater. So they observed clear increases in diversities when that community was exposed to 30% of wastewater treatment plant. On the other hand, in the right figure, you can observe how actually the degree of similarity decreases while increase the concentration of this wastewater in the experiment. So you have here communities really similar each other with 0% of wastewater, and then that communities start scattered and being far different while you increase the level of wastewater treatment plant. So literally, these are just a really brief example about how we are able to understand changes in biodiversity using eDNA, in this situation, sample or specific treatment coming from wastewater treatment plants. But at FRAM, we are also conducting several studies, as mentioned early Daniel in the introduction, using environmental DNA in order to understand changes in biodiversity. These studies are conducted in 3 different countries. from different continents, and each country differ in their social, economical, and also more importantly, environmental development. We are interested at FRAM and also in my research to link all these biodiversity changes to what is understood the pollution fingerprint. So we are able to define a pollution fingerprint, a list of chemicals present in the environment, and then we assess how that chemical affect the biodiversity of any community in a water sample, sediment sample, mainly. So, for instance, in Sweden, we have two case studies, one in the western Swedish coast that is about to study the role of wastewater treatment plant and industrial facilities on the biodiversity of marine microbial communities. So we have in northern part of Gothenburg in Stenungsund, one of the largest Scandinavian industrial hub there. And we were able to collect sample 1 year and a half ago already, and marine samples, water samples, in order to conduct that study. We are still processing that information, and hopefully soon we will have some news about that. We have another study in south of Sweden. This study is about pesticides in a freshwater system, and we are also collecting environmental DNA from small rivers and a small stream running through intensive agriculture areas in Sweden. We have another case study in Kenya that is in the same line with the pesticide application as well, and how the community responds to that pesticide stress. But also we are interested to explore the environmental DNA as a tool to identify certain diseases quite relevant in the context of the Kenyan society. And last but not least is our case study in Chile, in where we have one case study in the central area of the country. It's a freshwater study in where we are characterizing again environmental DNA from a system with which feature a high level of mining activity, one of the largest mining operation of the country, and also really intensive agriculture areas there. And then our last study in Chile is in the northern part of Patagonia, and then we are focusing literally on environmental DNA, but also environmental RNA connected to chemical derived from agriculture activities. So we have several case studies in FRAM, all of them linked through the use of environmental DNA, and also use this technique as an approach to understand the biodiversity changes in the environment. But in this seminar, I will mainly focus and I will present you really selected results from our case study from Chile, and particularly the one from the freshwater system. And I will, this freshwater system, the River Aconcagua. The River Aconcagua, like I said, is located in the central area of the country. That area features a Mediterranean weather, and this river basin is considered by local authorities under a really high water competition. The water framework in Chile is a bit complicated in the sense that this water is privatized. So actually, any person or company or party can claim certain level of water use. So therefore, those river basins with a certain flow of water, you can claim certain liter per second of water. So therefore, you have the mining companies, agriculture sector, but also the city halls claiming certain volume of water for drinking water for the population. So it's a really, really high competition for water there. And of course, we are interested to see how metals and organic chemicals may influence the diversity of microbial communities, in particular in this river basin. The strategy behind was to basically characterize the river basin, collecting sample in reference areas, so literally areas with really low human development, so sampling site lost in the middle of the mountains, really low activity there. So we have 3 of them, and 1 actually in the middle of a national park. We also have 3 sampling sites in the main course of the river, and we have 3 sites in the agriculture area. So this site actually, there are 3 tributaries that run through a very intensive agriculture. So literally we wanted to have a quite diverse sampling strategy, able to identify or to link those sites to a specific pollution fingerprint, but also to have enough environmental DNA to test our working hypothesis. So water samples for chemical analysis were collected using this on-site large volume phase extraction device. This is in the picture here. And that machine is a portable pump with a portable cartridge able to absorb organic chemical from, in this case, surface water. That machine is also working very well for marine sampling, so we are using it in all our case study. Once we collect that sample, we screen for about 700 chemicals, organic chemicals, using a specific machine called liquid chromatography high resolution mass spectrometry. But also we recorded in each of the sampling sites nutrients and several physical chemical parameters. So that allowed us to have a baseline understanding of the chemical characteristic of each of the sampling sites along the river basin. Now, from an environmental DNA perspective, we collect eDNA from 3 different environmental compartments. So fresh water, surface water, biofilm, that is like in the photo here, you can see all these greenish organism growing on that solid surface, and surface sediment. So we have 3 different environmental compartments, and the idea was to try why we took from these 3 different compartments, because we wanted to understand how is the dynamic of the whole microbial community. If you would stand literally in the river, and then you will think that you are exposed to different chemical stressors. So as many environmental compartments, we think that is better for this characterization. We followed state-of-the-art library preparation, literally very normal and timing approaches for extracting the DNA and preparing our sample for the later data analysis. I want to briefly introduce you our workflow. So we took 3 different environmental DNA sample, like I mentioned before, water, sediment, and biofilm. Then for each type of sample, we use a specific column-based DNA extraction kits. Then we amplify that DNA using 2 different set of primer. One here, the 16S, aimed to amplify for what is understood bacteria organisms. And then the 70S is meant to amplify microeukaryota DNA. Since the time is limited and the result is quite long, I will just show you today the result about bacterial community, so 16S. All bioinformatics was conducted using the CHIMI software, with here I highlight some main filtering parameters, and we use the approach of operational taxonomic units, in where individual sequences were clustered together according to certain similarity threshold that was 97%. And then we use specific database in order to get the taxonomic assignment, so literally the name to which species that sequence belongs. That data was later on exported to our package, so a software that allowed us to do downstream analysis called PhyloSec, and in where we did all this community composition study, alpha diversity, beta diversity, and then we did some statistics in order to test if our environmental variables, like nutrients, metals, organics, antibiotics, are able to explain the observed variation in the environment from our environmental DNA data. I would like to highlight that we have made all our sampling and extraction protocol available online. They're open access. You are welcome to visit that link, and you will have a detailed explanation about instrument use when you want to sample water eDNA, which are the protocols in order to extract the DNA from those filters in the picture. And we made some modification in order to the traditional extraction kit in order to enhance the yield, so the performance of the kit. So you are more than welcome to have a look, and hopefully the information is useful in your study. We will also, please stay tuned, because we will upload soon another protocol about how we do all the workflow from a bioinformatic perspective. So literally how, or which are the key steps that you should pay attention when you want to analyze your data. So, well, this is the first general picture about organic chemical measure in the River Aconcagua. We cluster, or we grouped chemicals in different chemical classes. So you have biocide chemicals in gray, you have industrial chemical in orange, and pesticide in green, and pharmaceuticals and personal care product chemicals in light blue. You can clearly see in the left figure that reference site 1, 2, and 3 were the sampling site with the lowest number of chemicals. So although there were reference sites with really low or even sometimes no human activities around, you are still able to detect and measure few chemicals in the environment. We observe a high number of chemicals in tributaries 1 and 3, which is a bit expected, since you can see here in green a big bar connected to pesticides, since those tributaries run through agricultural areas. So it's very clear here the contribution, the runoff contribution. And also then we have our riverside number 3 with a high number of chemicals. That since if you think about the river basin, actually that side act as a final site collecting all the sample, all the chemical from the whole catchment. However, from a concentration perspective, what is in the right figure here, we did not observe significant differences in concentration in each of the chemical classes. So literally all the chemical classes were in the same range with no significant differences. However, we saw and we estimate that the reference site 3, located in the national park, was having the lowest mean concentration of chemical in our study. There is a huge downstream analysis about how we process this chemical information. I will not go in detail since the seminar is focused on eDNA, but this information was later on normalized to effect concentration and what is understood at the toxic units. And we use that information as a toxic pressure value in order to link that to our biological responses. So we did not link pure concentration, rather we use effect normalized concentrations. So this is the first picture of the whole bacterial composition. from different environmental compartments in the river. So you have here in the upper left the biofilm community composition, the sediment community composition, and the water community composition. This composition is organized at phylum level, so all the phylum present in our samples are described here in the right, and I just highlight the 4 major groups from a relative abundance. So you can see here that the protobacteria group of phylum in pink is present in water, sediment, and biofilm with a significantly high abundance in the water compartment. The second major group was the cyanobacteria group in green, which is very well represented in the biofilm compartment. It's still present in the sediment compartment, but a really, really low abundance, and in the same situation in the water compartment. So you can clearly see in the second figure here down how well represented is this cyanobacteria group, although is really, really low abundance in the other 2 environmental matrices. Similar story with the third more dominant group, the actinobacteria, well represented in sediment, and the Bacteroidetes, very well represented in biofilm communities. So that gave us a sort of a starting point to understand how is more or less the composition and diversity of our sample, and at least how the main groups behave. And from the main group perspective, our results are completely in line with other studies analyzing microbial communities and particularly bacterial communities in other freshwater systems also impacted by organic micropollutants. Of course, you can take all this data and then do a second level of analysis and focus, for instance, in rare species, or you can select certain group of organisms and then you can track their relative abundances as well. But this is a big picture about the major groups. Regarding the alpha diversity, which refers to the within sample diversity, we apply 3 different metrics in order to understand the alpha diversity in our samples. So we apply an observed metric, which relate to the number of species only. We apply as well the Shannon index, which reflect how many different taxa there are and how evenly they are distributed in your sample. And we also apply the COW1 or CHOW1 index, which consider species richness as a function of sample size. So literally, we have 3 different metrics. There is more metric available, but we wanted to test 3 different ones following different criteria in order to assess the alpha diversity in our sample. We also apply 2 different criteria here to understand that. First, we pull all our environmental DNA sample in categories. So all our reference sample were pulled in the category of reference site, river site, and tributary site. And what we did observe was that there were not difference in diversity when you do that. So literally, they were not different in diversity between referent, river, and tributaries. However, when you organize your data in another direction, and then you organize your data from a source perspective, so from where is coming actually the information, we observe clear differences in diversity between the different environmental compartments, where sediment, here represented like a S, was significantly much more diverse than the other 2 environmental compartments, biofilm and water. That suggests if we go 1 page back. So for instance, tributaries were having a really high number of chemicals, pesticide related. So our results suggest that overall organic chemical did not affect or alter the species diversities of this bacteria community in the River Aconcagua. If we move to another metric, which is the beta diversity, so it's the similarity or dissimilarity between our sample, we also conduct two different analyses. One was also based on sampling site in your left here. So you have the same group, referent, tributary, and river. And in the right, you have also the same sample, but analyzed following another criteria. So the criteria of which is the source of the data. So sediment, water, or biofilm. What we clearly observed here was that reference site cluster almost independently from the other two from the two other sampling sites. There were not differences between a river or a sample coming from a tributary. It's a different story when you observe or you analyze the beta diversity from a compartment perspective. And here, clearly, sediment stand alone, very clearly differentiated from the other samples. And we observe also an overlap in some samples here between the biofilm and water. So, and in order to understand this, to understand and to explain this observed variation in our sample, we conduct a statistical analysis called distance-based redundant analysis. That analysis allowed us to get a better insight on the role of our environmental variable as explanatory variables in our environmental sample. So literally, there are three key steps in order to conduct this analysis. So you need a similarity matrix. So actually it's our beta diversity information. Then you analyze this matrix through a principal coordinate analysis, and then you assess the explanatory power of through eigenvalues of each of these variables. So summarizing this, but it might sound a bit complicated, literally the analysis shows us that there are 5 environmental variables here. So you have silicate, phosphate, nitrate, and nitrate, all of these nutrients variable, plus antibiotics, actually explaining very well the observed variance of our sediment communities. And we can focus, for instance, in the antibiotics section, in where those set of chemicals, this is a mixture of antibiotics, actually explain very well the variation in sediment sampled from the Riverside 1 and the Riverside 2. This is very, very consistent. We conduct the same analysis now in the biofilm community, and the analysis shows us that there for this set of samples, there is 3 main environmental variables explaining the variation from our sample. And again, we can see here that antibiotics really, really a strong correlate here, explaining the variance of our sampling site R1. So what is consistent in our result is that antibiotics explain very well the observed variance in the Riverside 1 for both sediment and biofilm communities. Finally, we pull all our environmental samples. We took everything, eDNA from water, from biofilm, and from sediment in one analysis. And again, that analysis shows us very, very clear that the mixture of antibiotic measured in the River Aconcagua explain very well the variance of the Riverside 1 and Riverside 2 for biofilm and sediment, and Riverside 2 for our eDNA from water sample. And here, if you give a closer look to the rest of the significant variance, you can see that all nutrients, or at least those 3 nutrients, very well explain the variance from the tributary side. And just remember that the tributary sites were the one running through agricultural area, usually those are sites very well characterized with high nutrient load. So finally, I would like to highlight at least 3 points from our study. One, that chemical pollution by organic chemical did not affect the species diversity in the River Aconcagua. The composition of the sediment and biofilm was significantly affected by nutrients and particularly by antibiotics in our study. And finally, our results suggest a site-specific role by antibiotics structuring the community composition in the River Aconcagua. I would like, of course, to thank all our collaborators in Chile and in Germany and China. And you are more than welcome to follow us on our Twitter account and our website for future results.